I have been privileged to live in a number of beautiful places throughout my life. Here in Squamish, I am drawn by the view of the sunset behind the Tantalus as I finish my supper on the patio. It is a fascinating and highly complex biological system that allows us to connect to the beauty of places like this, the visual system. Evolution has produced an eye that functions like a basic camera, allowing organisms to perceive visual stimuli. Incoming light enters through the pupil of the eye and passes through the lens before contacting the retina, which is a piece of neural tissue that rests in the back of the eye. This light information gathered by the retina is sent to the brain through the optic nerve, and visual portions of the brain use this information to construct the visual world that we experience. If you shine a light to my eye and look through the pupil, this is what you will see. The retina resting in the back of my eye from a front-on view. In the central region of the retina is the macula, which contains a high density of photoreceptors, rods and cones, as you may be familiar, and is necessary for sharp vision. In particular, the fovea, occupying the very center of the macula, is responsible for the highest resolution of vision in the eye. Now, imagine if we dissect the retina so we can observe its structure more closely. You can think of this as though we were to cut a tennis ball in half and closely look at the rubber lining on the back of the inside of this ball. Now, if we dissect the retina in this way, you'll see that it is composed of multiple layers of cells, as shown here in an image I've taken of the retina. And the different types of cells in these layers within the retina have specialized roles in creating a visual image. When incoming light enters the eye, it passes through these layers of cells before contacting photoreceptors in the very back of the retina. When light incoming through the eye activates photoreceptors, rapid chemical and electrical changes occur that are sent through these layers of cells, out the optic nerve, and into the visual portions of the brain, allowing an organism to interpret and act on visual stimuli. In this way, the retina can be thought of like the sensor of a camera, whereas the visual system in the brain is the computer that processes and stores this information. We humans are visual creatures, with more areas of our brains devoted to perceiving visual stimuli than any other sense. However, the environment within the eye places the retina at risk for various types of damage, some of which can result in irreversible vision loss. Due to our dependence on vision, diseases that damage the functioning of this system can be devastating and greatly impact people's lives. Still, there are a handful of conditions that cause vision loss that the scientific community does not understand in considerable detail, and as a consequence, these diseases do not have a cure. Age-related macular degeneration, or AMD, is the leading cause of blindness in the elderly of industrialized countries, afflicting 1 in 20 people over the age of 60. At first, AMD causes central vision loss, and as the disease progresses, eventually, all visual abilities deteriorate. The causes of vision loss in AMD, however, are still unclear, and there are no preventative cures for the early stages of this disease. Therefore, to develop treatments for this condition, the molecular and cellular events in the retina that contribute to vision loss must be better understood. The disease progression of AMD is at first characterized by the accumulation of fats and proteins underneath the retina that form deposits called drusen. When these drusen deposits build up under the thin retina in the back of the eye, they become yellow and round in appearance. The images behind me compare a healthy retina, shown above, with a diseased retina that contains drusen deposits, shown below. Now notice that these drusen deposits accumulate mostly around the macula, which, as I mentioned before, is necessary for sharp vision. Interestingly, these deposits contain various proteins that are associated with other age-related diseases, like Alzheimer's disease. For example, a protein called amyloid beta, or A-beta as I will refer to it, has been studied extensively for its role in neurodegeneration in the brains of Alzheimer's disease patients. And this protein, A-beta, has been found in these deposits in eyes that have AMD. Recent data suggests that this protein, A-beta, may have an important role in vision loss in age-related macular degeneration. Over the last 2 years, I've collaborated with Drs. Trevor McGill and Martha Neuringer at Oregon Health and Science University to investigate the causes of AMD in monkeys, which have nearly identical eyes to us, and where this disease also occurs naturally. Most recently, our research has begun to focus on the involvement of this A-beta protein in vision loss through this monkey version of AMD. Before I continue with the story any further, however, let me first talk about how A-beta is made in the retina so we can discuss ways to study its involvement in this disease. Genetic information is stored inside of cells in DNA, and this information is the instruction manual for cells to make proteins. And proteins are the workhorses that allow cells to function. So DNA is the information, and proteins are the working stuff. Information in DNA is at first rewritten through a process called transcription into a messenger termed mRNA. These messengers travel to different areas of cells where they're then read to build proteins. And this process is generally what we refer to as protein expression. In disease states, however, protein expression changes and can have negative effects on cells in the brain or the retina. Sometimes certain proteins can be made too much, or they exist in areas where they should not be, such as in the retina, the brain, or elsewhere. This can cause cell death, like when the A-beta protein accumulates around memory regions in the brains of people who have Alzheimer's disease. The A-beta protein might also cause cells to die in the retina of eyes that have AMD. So when proteins are involved in a disease, we want to find out where they are coming from so we can study their involvement in the disease and eventually stop these proteins from damaging the system. Our group set out to detect the A-beta protein using protein markers that have fluorescent labels. When applied to retinal tissue containing the A-beta protein, these markers will stick to different areas of this protein, and their fluorescent labels act as a signal as to where exactly that protein is located. Now, this is important because it indicates what areas of the retina are involved in the disease or are being affected. And as I mentioned before, different regions in the retina have different types of cells, so we can learn something about what types of cells are involved in the progression of this disease. Okay. Using this approach, our group detected the A-beta protein in the diseased retina. In this image, a red A-beta protein marker and a green marker have been applied to the retina to show where exactly this protein is located. In the image here, the area with the round appearance is a drusen deposit, which is being labeled with the red marker, and the green marker is labeling photoreceptors, just above. So this is identifying the A-beta protein, this disease component in the retina, in these two locations. Right? However, the keen eye will notice something peculiar about this image. Both the red and the green marker are intended to detect the same thing, the A-beta protein. And we should therefore expect them to appear in the same location of the retina, kind of mixed together, their fluorescent colors. However, in this case, we are seeing the two colors identifying the A-beta protein in two different regions of the retina. And there's not overlap of these fluorescent colors, and we couldn't make sense of this. So, in general, when you get an unexpected result like this in science, one of two things may have happened. Either you messed up, or you discovered something. I, of course, am hoping that the latter of these two possibilities is true in our case. So, the question that followed for us was, how is this happening? Where is the A-beta protein coming from? Because that is not clear based on this image. Okay. Well, what's another way that we can go about to figure out where this stuff is coming from? As I mentioned before, the A-beta protein requires a preceding messenger to carry genetic information to the area of a cell where the protein can be built, called mRNA. What if, rather than detecting the protein, this disease component that we're interested in, what if we could detect the messenger, this precursor to the A-beta protein, to reveal the initial source of where this thing is coming from? Well, I've made a molecular tool called the riboprobe, which does exactly that. Riboprobes are able to stick to mRNA before it is translated into protein. You can imagine this functions sort of like a piece of Velcro. Fluorescent labels are incorporated into the riboprobe to indicate where exactly this messenger is located. And that's important again because it indicates what areas or cells are involved in the disease in the retina. were causing vision loss. So our group is currently in the process of applying an A-beta riboprobe, which I've designed and synthesized here at Quest, to a diseased retina to show where exactly the A-beta messenger is located and what areas of the retina are producing this A-beta protein and are involved in the disease. So we expect that a layer of cells in the retina called retinal pigment epithelial cells, or RPE cells for short, will make this messenger and might be responsible for the production of the A-beta protein in the retina. And other groups have found this result in rodents, and we think this would make the most sense of our present data. Because if you think about it, RPE cells, they sit here, are located between the two areas where we have found the A-beta protein using these protein markers. So maybe the messenger is coming from the area just in between. Preliminary data that we have begun to collect indicates that the riboprobe works. It's able to find the A-beta messenger by giving off a fluorescent signal, and that RPE cells here do in fact make this transcript. However, our preliminary data suggests that more of the retina may contribute to making the A-beta messenger than just RPE cells. So the picture is more complicated than we thought. In any case, whatever we discover in the coming months of our work will likely lead to further inquiry about this aspect of AMD. The goal of this current research is to identify where this disease component is coming from in the retina, what parts might be involved in vision loss in AMD through making this disease component. Questions will remain in the future, though, about how this protein, A-beta, actually causes vision loss. How does it impact the function of the retina? And other questions might remain about how A-beta interacts with other factors in this complex disease that cause vision loss as well. And finally, if further research supports that A-beta is in fact involved in visual deterioration, how can treatments be developed to target this contributing factor to vision loss in AMD? But why am I doing this? Why do I care about this? Well, I value the ability to connect the visual world around me. And we are visual creatures, and our daily lives depend on this vital sense. When diseases such as AMD take this important means of connecting with the world away from us, it can be devastating. I want to make a difference for individuals afflicted by AMD through determining what factors contribute to vision loss in this disease. Finally, I want to thank Drs. Neil Melvin, Trevor McGill, Martha Neuringer, Jonathan Stoddard, and Dr. Richard Wildman for all their great help with making this project a success. Thank you.